so here's two key resources, URLs to enter either your memory banks or put it on a browser on the side of your window. So what I'm going to go over is what's been done in the genomic characterization of embryonic stem cells and induced potential cells, and getting them to differentiate and then characterizing them. And somebody's knocking on my door. Carolyn Trace. Okay. So
Rasmussen.
In the conference.
Proceed, and then get some orientation here. The basic goal of this genomics database resource is to give a consistent handling of multiple cells of origin, two comparisons at the transcriptomic and genetic
join the conference
level between these different IESs and human ESCs, and get them to differentiate out a couple of different directions, and then do comparisons of what their differentiative potential is, and then what are their molecular signatures, and all that data together. What can we learn about pathways that are critical for a healthy pluripotential cell, for differentiation pathways, mechanisms by which differentiations occur and maintenance occurs, all that. So we do a consistent characterization of all of the cells and different
join the conference.
We can spot key aspects of molecular networks responsible for pluripotential cell biology. Okay, so how's it looking? What are we doing? In the cell characterization core, we should probably just go over that workflow. Carolyn, let's go receive cells from the different hub labs, and that were used by various mechanisms, plasmids or lentivirus or mRNA or sort of thing, or from the intercell mouse, and have been gone through our procedures out in the hub labs, and then undergo standard culturing here in Cincinnati, and then RNA. Then they're characterized by factor markers, growth and differentiation rate, any irregularities in their histocellular morphology, as well as the CNV analyses to see if there's genomic irregularities. Then and then they're profiled molecularly through RNAseq, DNA methylation, and microRNAseq. And so far, the RNAseq that's been done has just been single N 50 base. And we're in survey mode for what are the key things going on across the different samples, and are there clear outliers in those samples. And then we decide on samples that are highly representative of the particular subtype that we've looked at from cell of origin or differentiation state, then we can use deeper paired N100 sequencing to get a much more accurate view of the transcriptomes in each of those samples. We haven't started any cells for the paired N100 profiling yet, and we're still trying to see how the data is all lining up, and we have to do to come up with a really robust
Adam Margolin
has joined the conference.
Hi, Anna.
So,
okay, so given all these samples and those 3D characterizations, although I say already that the RNAseq is not particularly deep at this level, we're still in survey mode, and then the phenotypic characterization, how do we start to put it together into a database that we can mine and get some useful information content out of, and tell something about any of the methodologies that are at play here, and what do we need to do to get more out of pluripotential cells. Understanding their mechanisms to get them in a healthier state, more efficiently obtain them, and then use them in experiments that are modeling diseases out lineage-specific directions. So we really have a resource that helps learn about stem cells and their potential and their differentiation products. These are the basic samples then that are getting fed into this analysis pipeline. Then you've got the embryonic stem cells and the iPSC, the induced pluripotential cells, and I talked about the different cell of origin effects and method of induction effects. Those are all underlying variables that'll trace into the iPSC, and for each of those, they're grown under standard conditions. Right now, our standard conditions are room air, and that's
has joined the conference.
5% CO2.
5% CO2, but the oxygen is at 20%, and that's something that's in the air for possible modification. So the EBs are the standard
has joined the conference
that Carolyn uses is a 20-day culturing with the FCS and what's NEAA?
Non-essential amino acids.
Okay. Plastic.
Gelatin-coated.
Gelatin-coated plastic. Okay. And then the endoderm, mesoderm, ectoderm protocols are short.
Has the conference.
Be driven by activin A and reduced bovine serum and plus or minus BMP, FGF, and then what's B43, 543?
Those are small molecules to block TGF-beta and BMP signaling, I believe. I can look at those, but they're small molecules to block these endoderm differentiation.
They're relatively, but we're still experimenting really essentially with
left the conference.
Comparative conditions. So pipeline for each of the different data types involves, and right now we're still, as you may or may not know, we're transitioning to a more enriched bioinformatics, more automated standard pipeline infrastructure through the phage synapse bionetworks system, and so we're all really excited about pushing all the data through that. Right now, we're still making those connections into it to make it work reproducibly. But so what you're going to see so far is what is done with just essentially one particular pipeline, which I'll go over in just a second, on the top hat cufflinks cuff-diff pipeline, and really most of the results I'm going to show are against the ensemble gen code models reference. That makes a difference about which gene forms you see. For example, Sanders and on would see slightly different versions of gene models and splice form models and that sort of thing. And then there's also another when you run gene models, you can collect everything per gene, or you can break it out into known and discoverable isoforms. And so all those different iterations that give you the potential to see, give you quantifications of separate isoforms of a given gene product are.
This is Jenny. I had a question. When it's a new genome build, how easily can all the data be run against the new build?
It's going to be a snap. But right now it's a whole hour process to or more to get everything, all of the there's two issues. One is is it a gene model change, or is it the underlying reference genome change? And if you go from MM9, or I'm sorry, from just
Carrie Bell, NHLBI,
has joined the conference.
The default reference 37 human genome to now a reference genome that includes alternate scaffolds and haplotypes. That's to redo all of your alignments to genome. So that takes longer. So you basically have to regenerate the BAM files. But if just the gene models change and the alternate splice form detection algorithms change, then that's using the exact same alignments against the reference genome version that you used last, and then you requantitate per those different gene models.
Okay, thank you.
All of those kind of results, we'll see that in kind of a tree structure of that relates to these different alternate pipeline views, and then also really for the people who want to get brave about it, you will be able to interact with the Synapse website.
Has joined the conference.
To spawn a new analysis based on your either a library reference gene or gene model set, or your own. You can up and then run. Adam, would you say that's a good description, or? Okay, let's move on. Okay,
sorry about that. But yeah, good.
Okay, so for really all these kinds of comparisons, and so miRseq goes against miR being the same thing, Jenny, that miR-base is also frequently updated, and then all of the underlying reads that were generated in that data set can then be rerun against the new versions of base, new link RNA gene models, and that sort of thing. And so that everything from the raw data to the intermediately processed format data to the finished kind of normalized files per a given pipeline will all be there as like separate objects that you could just download and then use them locally, or you could use them from the NEPS website.
Follow-up question. Sorry to interrupt you. So one assumes that researchers are doing things and they do analyses, they publish papers, and they want to be able to cite which exact data set and build they use so people could replicate it. Is there a consensus within PCBC or within this infrastructure of how that would be done?
Yes, that's an issue that we talk about all the time, and we're really happy with the way that it's going to get done in the this pipeline.
Sorry for asking something that.
It's called the provenance documentation, and it goes over both the the versions of the reference genome, the versions of the gene model, the settings on the scripts, and then the version of all the algorithms. All of that goes into a single document that you can then print out and then save as a stored object, and then you can even back to it in see as it lay as you pull out your results.
Cool, thank you.
I can add a little bit. This is Larson, actually. So the other thing is you also get a DOI, which is citable by journals and generally acceptable by them as well.
True, for the specific build that you used?
You get a specific for the data that was generated, and then that will point you to a specific build that also has a DOI.
Oh, very nice. Okay, right.
Specific executable that also has a DOI.
Yep.
Has joined conference.
Thank you.
Okay, so then the same for miRseq, and the DNA methylation the methylation is a little bit, it's more static on the underlying data. It's basically this 450K CpG chip from Illumina, Infinium chip, but what does change are the relationships of those methylation sites to known gene promoters and epigenetic regulatory regions. So that is where several more complicated steps past what the annotations really that Illumina supplies for each of these CPG sites in the 450K chip.
Has left the conference.
And so anyway, as those annotations change, as they will for everything, the MERS, the RNAs, the alternate splice, everything is going to be constantly improving in what we understand about it. And so that will all be
would be better if you put it into something more penetrable to the water, because you're cutting for just an hour.
Oh, that question, I guess. So I'm talking without being muted.
Just check the third drawer, maybe.
Okay, so let's go on. Here's what happens when after you align reads to genome. You see here's two genes side by side, TCF19 and Tau5F1, and this just shows individual tracks from individual samples. You've got definitive endoderm here. Can you guys see my pointer? I hope so.
100 base pair.
Mark, I think you're not muted.
1.2 to 1.5%.
And okay, so then now we've got no difference in EB and definitive endoderm with respect to Tau5F1 expression, and those two samples show no expression of TCF19, whereas the embryoid body samples over here, great expression of TCF19, which is adjacent to the Tau5F1 locus.
Has joined conference.
So those you know that the RNAs are good essentially from each of the samples, because the other genes they have this reciprocal regulation pattern, but there's absolutely no reads coming from TCF19 in the you see this is marked definitive endoderm, embryoid body. This is the particular sample. It was this stem cell in 2011.
That's an accession number.
The 008, yeah, and then it was differentiated into embryoid body. So that's how you read this. If there's no suffix to it, then it was just a stem cell and the IPS state. That's our convention. And so, okay, so let's move on. Now what you want to do is
conference.
All the currently 139 samples in the database, you want to sum over these different genes and then come up with a normalized, just a raw expression table that's normalized per sample, usually for the read depth for each one of the samples, and then start to put it together and see what are the patterns of genes expression that are going on in the different sample types. And you can also see splice forms. I just show a really
has left the conference.
Cool. Well, no, I'll get to it later.
Join the conference.
On the day of the individual samples is Phil Dexmer's built a pipeline that lets us look for 5', 3' integrity of the RNAs, and does looks on the 3' end and the 5' end, and so we try and so we'll exclude samples that fail from this regards, that if less than 10 million reads, if there were less than 75% of the reads equally aligning to the genome. This is using TrueSeq, so it's OEA.
Has left conference.
The ribosomal reads are low. For this 5' to 3' ratio, and so this was a bad sample. And you can see what's going on here is that in this case, both the 3' end and 5' end, the 5' end you had reduced coverage, looking out from the 5' end, and then come to the 3' end coverage, it dropped down. So these here that shows little profiles were samples that we excluded in the database. So this is really technical artifact, and that's different than biological artifact of the differentiated state of the cells. We do want to have as many biological artifacts as possible so that we can see what different patterns are of outlier samples, but we don't want to see just technically crappy data. So those samples are excluded from the database before you start the build of the overall signatures. Okay, and this is just a summary of the top hat cufflinks pipeline and how it starts from known transcripts and after going genome alignments and then it goes through this process where it creates a final GTF. It's a mix of both known models and de novo forms that are found. These are all sort of configurable options in the cufflinks pipeline, the cufflink cuff merge pipeline that using that pipeline will be able to iterate over different settings of those to more be sensitive to finding different kinds of de novo genes or genes that are poorly understood but well documented in our data. Okay, so in trying to start to build a normalized expression table, what you want to do is and this is true for both the RNAseq, the microRNAseq, and the DNA methylation, is you want to start to build a table that looks at all the samples that you have that you think are decently measured, and puts them into a form that you can then start to do statistics on to look for analyses. And this is just the considerations that you have to make in allowing samples into that final table, and which entities of the 30,000 entities or more. If you're going to use alternate splice models, you'll have 80, 90,000 entities are in each row of this table, and one of those entities is you have 5 different entities from tropomycin 1 according to which promoter used and which exons were included or excluded, and you can get different counts on those, but we'll get that sort of stuff later. And so there are just criteria for inclusion in that initial matrix for the analysis. So I'm just going to show you what that looks like as you put it together. So here's a map at the 17,000 gene level. And so technical outliers were removed, but all the individual forms are in here. And so this is essentially without any statistics to say, I really only want to see the genes that show differential expression per these differentiated types that are listed down here on the bottom. That was not done. And so this is what you're seeing here is for the top 17,000 measured genes of the data. Now just using a blue-yellow being high scale, going on with the patterns of variance in the overall data set. So you can see really solid signatures in the iPSC, ESC. The iPSC actually includes ESC. I'm sorry I didn't label that. And there's some weird sorts of fuzz variations in the that are dividing different of the sample types within iPSC. And so sometimes you can even see some large group of overexpressed genes. We'll get to that in some more detail in a little bit. And then, but this, when you just sort of look at all well-measured transcripts and you come up with a normalization scheme that lets you just see the basic patterning, then it really helps you get oriented about what are the basic things that are going on here across these different types. So what one of the first things that that you see next to the stem cells. So what you'd say about the stem cells is that en masse there's thousands and thousands of transcripts that are in pretty good agreement that they want to be a strongly expressed transcript in the iPSC and the ESC, and then in the blue you can that as being that there are plenty of transcripts are abhorred for their expression from iPSC, and of those, good fractions of them come on in differentiated lineages that come out from the iPSC. And yeah, right, there's these different lineage samples here. So that's here, 8,000 transcripts maybe that are expressed
Maria Mirocho has left the conference.
By these lineages that are not by the iPSC, and some of them vice versa. Here in the EB, you see the extinction of thousands of transcripts that are well expressed by the iPSC. So that you can get a sense of the enormity of the change in the genome state going from iPSC to EB in this kind of a view. And then just as a fine point, when we're looking at these so EB is that's the longer plastic low driven differentiation. All get extinction of all those iPSC genes, but on the other hand, that's not seen in the definitive endoderm, mesoderm, ectoderm sample. The ectoderm is definitely more differentiated than either the meso or the definitive endoderm, but some of the properties of the definitive endoderm and mesoderm look like they're not that different. There's just a little bit of a single couple agent slight change of in the meso differentiation protocol. Also, the times are not very far in the differentiation. So what all of this stuff, this gestalt, then shapes your really think should be a reasonable test of statistical differences between these samples that let you focus in on different areas. So so one thing that would be you would want to use a general guide map to then thinking about where your really strongest differences are going to be. Namely that there's going to be a huge signature of genes that are activated. It's not huge, but it's in the hundreds of genes that are activated across the board, really, in DE, meso, and DE and meso that are not so activated in ecto, that are unique from the stem cell, but on the other hand, from the pluripotential cells. But on the other hand, many iPSC genes that are not that turned down by the DE. So when you're thinking about the biology of DE meso versus iPSC, you've got to consider that its active genes actually are also quite active in iPSC. And so what you're doing is you're shifting the PSC pluripotential cell now as a result of turning on this small signature, diminishing the signature of some of these major iPSC genes. You're also using a whole lot of the memory that was at work in the iPSC. And then a lot of these lineage specific genes up here on the top 5 are not particularly associated in the DE meso. Makes sense? It's just these are kind of orienting concepts that then you can use in more focused ways down the line. Let's go into just some of these signatures a little bit deeper. Now this is a look at the 5,000 genes which are most differentially expressed between EB and iPSC. So here you really don't see, and then here I'm showing the ESC versus the iPSC. So you really don't see much of a signature difference between the ESC and the iPSC in any level, really. But this is particularly now. This was a signature built on some difference between EB versus stem cell. So stem cell of either type, ESC or iPSC versus EB. So it's a nice kind of iPSC versus differentiated EB signature, and then you're kind of using that as a barometer of what is. Are there any type differences that come out in the ESC, iPSC? And the answer is really not. It's not certainly not a major component. On the other hand, here's your
soon. Having the conference.
So let's go on. So when you take a gene expression signature and you do analysis, there are lots of different methods for doing gene signature analysis. You can use the entire vector of all genes expression from the very highest to the very lowest, or you can look at the tail of the distribution for the genes that are most strongly differential and and so all that information about what's not there in the signature and what is there in the signature go to shape the biology of the thing that you're trying to study. So let's study into the stem cell a little bit. So here's that principal stem cell signature, and remember that because I told you that there's a huge number of genes that are also active in the definitive endoderm as well as the iPSC. Is it really a fair test to say that, oh, and then the rule is that they have to be strongly downregulated in DE? Well, you're probably, if you do that, then you're going to exclude consideration of a lot of genes that are continuing to be strongly expressed once you undergo definitive endoderm, the initial stages of its differentiation. So then it's you can find the genes which are most specific and most exclusively almost active in the iPSC.
Has joined the conference.
But there are actions with the genes of that you excluded because you used the consideration that it had to also be different, more significant, much more expressed in stem cells versus DE. You're losing the connectivity of these genes with those genes. And it's a funny argument, and sometimes it's useful to include and exclude those. And really, it depends on what you're going to be after. But that's the kind of logic that needs to shape your approach to mining data out of these signatures and trying to understand the biology of the cells and the differentiated processes. So here's that signature, and this some information content around those genes, which are in the disc now as being most stem cell specific. So tremendous enrichments for, and this is analyzed on top gene, and I'm going to show you top gene in just a second because it's important to see how now the PCBC data has been entered into, or at least the current version of it has been entered.
Join the conference.
So this is how your ways that you can get access to using that data right away without having to crawl through all the tables, although you should definitely do both. But okay, so here's this top 2,000 genes that are IPS specific, and so what's going on with those genes? Plenty of cell cycle associations, plenty of non-coding RNA gene associations, lots of genes devoted to modifying the state of chromatin, silencing and action, overrepresentation of genes associated with condensed chromatin, all make total sense. So those are the simple gene ontology associations. And let's say you want to attack chromatin regulation, and then really try and build a network view of what is the state of chromatin regulation in the iPS cell. If you brought together every single piece of information known about every gene, and then took this gene expression signature, how would you build a network that could best represent your view of the of chromatin biology as used in the iPS cell to manage genome? You'd essentially be clicking off these different boxes. So in the term in query is that's the number of genes that were from this top signature, or I'm not sure what I used. It was around 1,000 genes actually. How many of the terms in that express signature query have been annotated with any of these Go terms? There's 146 of the genes in the query had a cycle phase association, as compared to there being 882 genes in the entire genome that have cell cycle phase as one of their annotations. And then here's a value for that representation. But when you're talking about a given signature, we probably need to have just a tiny bit of considering of when should you use false discovery correction and when should you not. You want to try and understand the overall biology of a given signature. Yes, you need to use false discovery. If you want to understand key elements of a given signature that meet up with categories of high interest to you, and that those categories aren't necessarily an encompassing explanation of the overall signature, you have to take the false discovery off. And so you want to understand all of the sequence-specific transcription factors in any given signature. It's unreasonable to think that unless there's a PL5 with false discovery that I should see all my transcription factors. No, but what you will see are the transcription. If you turn it off, you will see all the transcription factors that are there, but you will definitely not see the transcription factors that are there if you turn false discovery on. So it's a funny point, but it's very important if you want to get lists of genes along a category and really start to understand that category of biological or biochemical function in a given context. Okay, go on. Here's just some other associations to mouse phenotype, and you can see means that when knocked out, a mouse phenotype association means that when a given gene is knocked out, there's 118 genes out of the 7,000 mouse gene knockouts that have been done that have been fed in the manuscripts associated with those knockouts to give abnormal cell nucleus morphology, and there's essentially a quarter of all those genes are in this signature. And then the same having to do with these embryonic lethality before even somites form. We're very overrepresented in that. So that's a nice sort of progenitor cell biology thing. But exactly what those genes are that are doing that could be divided out over many different biochemical categories. They could be dealing with cell-cell interaction. They could be dealing with chromatin control, cell cycle control, all that stuff. They're going to be probably overrepresented for cell cycle control in there. But there's the idea that you can have multiple genes from different categories that contribute to preventing a given essentially a disease phenotype by their mutation that are coming from different categories of function. That's where systems biology comes in, where we want to try and promote everyone's ability to pull together multiple pathways to better understand disease processes that are critical for development. Okay, just a few more things. Protein-protein interactions is in there, and so here's 60 genes associated with MIC interactions or simulation or IKEA, cyclin kinase, 2F binding sites, MIC binding sites, that sort of thing. No surprises, but you want to have these connections there. This is essentially, these are the things that you can use in building networks to understand the activity of the different groups of genes. Here's this. This is from MSigDB. It's called the co-expression tables of MSigDB, and from the Broad. So this is that top EFPS signature. Then it only got 208 out of the 378 genes that were said in a meta-analysis by Ben Parath et al. That these are genes are said in 5 out of 20 profiling studies to be overexpressed in human EF cells. So what that means is that that's cool that we got such a good take on this one particular signature. But there's two things that are going on here. One is we have 1,000 genes in the signature, not 200. So there's 100 genes that are outside of this circle. And on the other hand, we don't capture 170 of the genes in that signature for some reason. And it turns out that reason is going to be pretty interesting because if you go through the process of mining this, as I do on multiple occasions, what you'll see is actually that those missing genes from here are in definitive endoderm. They're in differentiated cells. And signature is actually contaminated with differentiated things coming out of IPS studies. And that, I think, to me, that crystallizes. And we can look at that in more detail, and we should, and we will, and we'll probably put that into the first paper. But it sort of crystallizes why doing things in this highly standardized format is absolutely critical, and instead of just sort of willy-nilly grabbing things from the literature where you think that the conditions are adequate to the answering the question. So it's a really big point. Okay, and then there's other interesting things in MSigDB about correlating with key regulators and other. So these differences from these stem cell signatures, the nanog targets. So actually, okay, look at the nanog targets. We've got 140 genes that have been said from BenPreth to be targets from chip on chip, and so that immediately gives you this ability to slice down the signature into those that are direct targets versus secondary targets. So that's what this does is it shows you how data from other sources can be put into the equation to help explain what's going on. And not only that, not just gene expression signatures, but also things like a DNA methylation state, a transcription factor binding site, an H3K methylation or acetylation state. Those things can now be structures that can be intercombined, and that's what we're going to let you do. Okay. I'm going to run out of time, and I want to show you how to interact with the data resource. This is just it's hard to set up the database, and in boxed in red here are embryoid body gene signature differences that come from this pink versus blue versus yellow, which turn out to be CD34, embryonic stem cell, and fibroblast-based cells of origin that were then asked to differentiate out into the embryoid body. And then we did a supervised test for looking for genes that are significantly different in the embryo body as a result of the cell of origin. So we'll have to get back to this in a little bit, and this is it's a complicated story. Jeffrey is part of is really behind this signature in Elias Zambidis' lab. That's part of this data set. Okay, so I'm going to just go through one quick little thing, and that I hope can link up to microRNAs if I'm really speedy. This is a definitive endoderm signature, and you can see, like I was pointing out, the definitive endoderm signature is a whole lot of the IPS signature over here on the far left, plus it's got some unique stuff on its own. And the approach that we're taking to coming up with separate signatures is we're calling it database shredding, where we'll pull individual clusters of genes. So this is now 800 genes that are part of it. This is a 2,500 gene signature. This is 800 gene signature, and it's just a k-mean subset, which are pretty specific for the definitive endoderm. And so leave this. I didn't have time to get the enrichment for this, but this signature is highly enriched for genes that are controlling the development of endoderm and mesoderm, and it's SOC17 is on the lead of it. But I'm going to flip very fast over to the microRNA data, and then I'm going to show how microRNA data can help analyze this secondary signature. We can link up those two things in a networky way.
Has left conference.
Here's microRNA data, and so this is a view of the top 490 microRNAs of the in the data set with some of the best samples that we have. So here are the on the right, so we're going microRNAs from left to right, and we have a signature of microRNAs that are very strongly expressed here in stem cells, and some of them are very downregulated, and some are slightly downregulated and going to definitive endoderm. some of the, over here on the far right, are some strongly expressed MERS that really go across the board, stem cells to even out to peripheral blood mononuclear cells. And there are significant differences between DE and SC with respect to microRNAs, but they're not as huge as you would think. But the that are different are really interesting. So this is a little bit more on the slightly different organization of that RNA signature. And the normalization on this is in proportion to the absolute expression level of the microRNAs. So microRNAs are really like genes too. They have mass action effects, and you absolutely need to know what their absolute expression level is to know what's their relative contribution to the biology of the cell. So in other words, a 10x change of a given microRNA that goes from 10 reads to 100 reads is very different than a microRNA that goes from 1,000 reads to 10,000 reads. And so just the molar effects. So this is a scheme that lets us look at relative expression of microRNAs in proportion to their actual expression level. So here's this is one quick, this is easy. MIR302 isoforms here in the stem cells have a massive downregulation into virtually all the other sample types, and this is well known, and not it's substantially down in definitive endoderm as well, but not nearly so much. So how do you use all these signature databases to kind of better understand in a systematic way what's the likely impact of MIR302 downregulation on these other lineages, and what does that tell you about what MIR302 is doing in the stem cells themselves? And there's all these different, you look at the microRNAs impact a system both within the system itself, and then as the system changes in its differentiation from state A to state B, and so it's going to shape all of the things, multiple considerations there. So you really essentially need to know about microRNAs that are turned down and RNAs that are turned on in going from A to B, and they help your reasoning about what could be going on. But this is a relatively simple situation where MIR302 is drastically downregulated. Its molar level of expression is super high. And so what it turns out if you just did an intersection of MIR302 targets, and we took that stem cell signature and asked how many of those genes of the stem cell signature are the targets of MIR302, turns out there are not that many. And instead what I'm going to do is take the signature from all the differentiated lines and I'll intersect MIR302 with that, and then you see a gigantic number of genes associated. And there's a publication out there from I don't know, a year and a half ago that gets at this a little bit, but the magnitude of it they completely missed. And I'm sorry if anybody's related to that group, but if you take the overexpressed genes of the embryoid body and the overexpressed genes of definitive endoderm, and you put them all into a bucket, and then you ask, okay, how many of those genes are connected to MIR302? I've done that, and then you ask, okay, what are those genes? And it's a who's who of transcription factors and transcriptional control, super rich in cis elements too, and it's all about differentiation in transcriptional programs. And so you've got gigantic. I'm sorry, I must have cut off the gene numbers, but it's very large numbers of genes that are involved in heart development, vascular development, hematopoiesis after the stem cell state. So it's not to really understand what MIR302 is then doing in the stem cell. You really need to look at this entire transcriptome that comes out of the stem cells after they start to differentiate. And so what the hypothesis would be, and that was made in this.
All the time, so it's going to be complete rubbish.
Has left the conference.
Those are targets that control lineage-specific differentiation. So MIR302 is suppressing that. so it's there's that part of the gain that you get by having both stem cells and the differentiated cells for comparison. And yeah, this on just on the gene ontology level on mouse phenotype, but yeah, here's a sense of what some of these genes are. All MIR3 targets, so it's really cool, and there's a whole lot to do more from that. And that's just the stuff that's relatively well understood. And so how would you look at some of those genes? This is what we're building into the system is going to be a way to pop out these both known and candidate pathway views that let you see for a given gene signature.
Left the conference.
Uh-oh. If I still leave the conference, could everybody do it all at once?
NHLBI
has left the conference.
So anyway, so this is how you can look at what some of those associations are. This is a set of genes, that MIR302 signature, that are involved in post-pattern specification, dorsal ventral, bilateral symmetry, and so lefty 1 and lefty 1 and lefty 2 are known to be part of that, but that all these genes here are MIR302 targets that are involved in bilateral pattern specification. Now you've got a much bigger network of genes to consider for MIR302's impact on the pluripotent cell. And so what you're saying in this kind of a view is that the targets of MIR302 that are suppressed in the pluripotential cell are directly involved in the suppression of developing left-right symmetry, AP, DV development.
So Bruce, this is Jenny. I have a question about that. And it's sort of coming out from the left field. So here you have a signature. How do you have a way of making these signatures themselves computable? So, for example, you have that signature right there. What if I had a different signature that I had generated on something different, make it up, I don't know what it is, and I wanted to be able to compare, say, my signature that I have in hand to this one that you've generated or to some other ones to try to figure out whether they're related or not. Do you have a way of doing that?
Yes. Yes. So let's head right now over to the top gene in Gattaca. So let's see. I'm going to make this bigger. Maybe more. So this is top gene, and so you go top fun is the link to use, and so let's just put in SOX17, and you put in a list of, you put in the entire genome list of genes, or you could put in genes or one gene. Let's just try SOX17, and here when you're just putting in one gene against all this information content, you take the false discovery. Okay, so now down here in Expression Atlas are a set of signatures from PCBC that 17 is one of the genes in that signature. And so it could be a very small list of genes, and so the p-value looks like it's really is more significant for the smaller list of genes. But sometimes what we want to do is really a fairly big signature of it, and then so this is the ratio of
has left conference.
This is a really cool thing. In mine right here that I've highlighted, the name of this gene signature is called ratio DE versus SC100. And over here is a more verbose description of what that signature is. It's the ratio of induced definitive endoderm to stem cells, and it's the top relative expression ranked by ranking method, and top 100. So there were actually 99 genes in that signature. So this is a DE signature from the database. Now you can take your signature and you can see then how does your signature from one of your experiments, Jenny, compare against that. And so this is, and you might see, oh, okay, that I've got 10. Let's do that for this signature. So I'm just going to grab that I click that 99, and then this is that signature. And now what I can do is iterate back, take the signature back into the analysis database, and this process again. And I'm going to use these settings here of supreme false discovery and cytobands and PubMed. And I'm also going to suppress in co-expression atlas and co-expression because that's the origin of this list of genes, and here too. And now, so now I'm looking at this 100 DE signature, and you can find what were the genes in that signature here by in that window. This is the list of them. It included SOC17, LefD2, a link on FGF17, all really good guys for what you are to know about. But how do we know that? And so now this is the summary of what the outputs are and the relative enrichments of stuff. So that in that top 100 signature, you have 16 genes known to be critical for gastrulation, 12 for endoderm development. Actually, it's not. That's more. Here, phenotype, you'll see that actually some of these categories exceed that which is in ontology, just showing that mouse phenotype associations to genes is, in cases, more up to date than gene ontology associations. And now, okay, this is a funny thing. So there's hard development in wiki pathways. That's probably a pathway that was entered by Nathan. Int is huge in this as well. But now go back down to the co-expression atlas, and now you can see how that signature from the PCBC data set compares from that was this endoderm versus stem cell 100, how it compares to other signatures. And so you can start to see places where, so in the top 1,000, it'll all be there, but now you can see, oh, it's part of a K cluster. So here's got, it's in the top 1,000, clustering it like I did, like I showed you that more refined signature. Now I've actually enlarged it to a 523 gene signature that is characteristic of what some of the top genes of the top 100 show. And so now you could connect the biology of those to, you've explained almost all of the 100 in this one more focused patterned expression. When you do just a ranked gene list from a cell type, you're ignoring the complexity of the expression patterning across the other samples in the set. Whereas when you do the K-means group, then you get out.
Has left the conference.
Just jump ahead really quick and show you one more thing on that. Okay, I'm going to be lost. Where is it? PCBC. Okay, this jumps ahead 2 steps. In the microRNA data set, there was one microRNA that was extraordinarily induced, MIR375. And there were about 4 microRNAs that were very strongly downregulated, coming out of the IPS to definitive endoderm. Those, and MIR26B was top amongst them. What we want to do is try and understand how the microRNA data connects to the gene expression signature. So these are all genes that are super strongly expressed in definitive endoderm, and many of them are also strongly expressed in the IPS in the stem cell state. But what the hypothesis here would be that the differential MER expression signature is going to have an interesting effect on the proteomic output of this transcriptome. So, so now dividing that DE signature into those genes that are the targets of MER375 and the targets and other things that they're connected to biologically, all the genes that are connected to the down-regulated MERS. And so what you can see is that what you're doing is now polarizing, and this is the involvement of those genes in endoderm development and sulfate commitment. So this change in the microRNA program is going to be, you're going to be inhibiting 6B and T3, and then you're going to be enabling the activation, the translation of SOC17 and GATA4 and EOMs, DUS6, etc. I hope so. So that's goes into how we're going to do network biology out of this data set that's going to help us really better understand lineage-specific differentiation and what are all the events that ensue. That, to me, summarizes a whole lot of what the potential is in this, in this genomic atlas development of this database that's going to really finally dissect lineage-specific differentiation. And so we'll be able to extrapolate back to transcription factors and microRNAs that could be, better enable direct reprogramming of cells out to specific lineages, and as well as let us model the effects of mutations in any of these genes in giving disordered development or end function, terminal differentiation function cells. So I guess maybe I should stop here and see if there are any questions so far, but I do need some follow-up.
Conference.
How to interact with the data, how to download the data. There's one more. There's just one more thing. I wish I had left more time for it, but there's a link that I gave you in the very beginning. That is P. So right here, this is the beginnings of the portal. It's gaca.tchmc.org/gaca/pcbc.jsp. And so what you can do now is search into the database, and these are like different categories of information that we have available so far. And so here's this expression atlas, and you can do things like you can take individual signature. So here's one signature, which is the ratio of embryoid body blastocyst to derived from blastocyst versus embryoid body derived from CD34. So it's top 500 genes which are differential there, and then you compare that to let's see. Another one. And so you can start to set these things up in Venn diagrams, and then it has multiple gene signatures at once. And so you'll easily be able to do that for the mRNAs and the transcription signatures, and you can export it and you have to check them. This is a more intense process, but also down here, let's go down in this now from mouse phenotype. So there's 108 genes that are known to give you abnormal blastocyst morphology, and then if you can now see in that there's actually an intersection of just one gene, and this wasn't. Sorry, I didn't have a more better query. But basically, this is now how we can intersect mouse phenotype genes with the different transcription signatures, and then you can sort of zero in on what that gene is, or what the intersection is. Click that, and then it appears here at CDX2 and sort of thing. Down here in there's gene ontology categories having to do with development, and so you can say, okay, this is relatively simple. Take the blastocyst development category.
Margolin has left the conference.
Take that as a gene set, and then how does that gene signature get does it appear in the PCB data set? So now I take this as every gene associated with blastocyst development, and see how does that signature appear in the PCB data sets down in this category of expression atlas. And it turns out that that signature is very well shown in primitive germ cells. This is a cell type specific atlas data, so it's a mix. You have to go down to where PCB data starts to appear, and so the first place that list of genes appears is in this mesoderm induced from S cells.
And Bruce, the co-expression, does it include both positive and negative?
No, it's always the first one. It's what name is. So in this case, this is the top 100 genes, the K cluster number 1 from mesoderm.
Okay.
But if it says something versus something, so here it's DE versus SC, now the top 100 genes that are higher in DE versus SC, and you still have 4 genes.
Okay, now that's a interesting. So it's the top 100 genes that
between definitive endoderm and stem cells.
Okay.
So that
are expressed, or the top strongest expression, either positive or negative?
No, it's always, so you think versus is probably not accurate. Nathan's made that point too, that we should say overexpressed versus stem cell.
So I guess that's a scientific question. Is the
left the conference
expression equally interesting or valid, or how is that pulled out?
That is pulled out by the reversal of these terms. So we would look for a list that says stem cell versus definitive endoderm top 100. It's automatically generated the top and the bottom. It's just differently.
I'm going to have to go soon, so my apologies for leaving. I think it sounds really interesting. I'm thinking that it might be really helpful to get this in the hands of the researchers as soon as possible.
This is all there to be used right now.
Sometimes they need help.
Yes.
So what is the plan? I know that there's bioinformatics expertise. I know you guys are all much more than just bioinformatics weenies, right? I know there's bioinformatics expertise at each of the centers on this group. So are you all going to go out and proselytize at your various research groups and home institutions, or how are you going to get the rest of the PCBC Consortium over the hump of starting to use this and get familiar with it, so they can start kicking the tires and telling you how it does or doesn't serve their needs?
Important point. It's budgeted
is Nathan, and I think for me, as even a novice user for this data, and I didn't even know a lot of these signatures were in the co-expression list, and I'm just going into TopFun and just doing some analyses, I found a lot of interesting PCBC associated hits. So I think number one thing a center can do is just go use TopFun. I mean, just take a list of genes and start playing with it, and you'll start to see signatures come up that are related probably to what you're doing. I think that's kind of the easiest, fastest way for people to jump into it.
But Nathan, this is Michael. I think what we need is a training session so that fellows and junior faculty learn how to navigate this system and get some examples and workshops in walking through it.
Yeah, I agree with that too. I was just as an interim kind of a step, I think that even just jumping into this tool, I totally agree. I think you need some more in-depth tutorials for this, but even just navigating this on your own really gives you a lot of moments in terms of insights. If you just start playing, it's a really easy interface to use if you just start playing with it. But if you don't have data, then you definitely need somebody to walk you through it.
And we're going to have direct to network generation buttons added to over the next year, and so it's going to be, and then all of these screens. This was sort of a semi-automated crude shredding of the initial data. It says it was a couple months ago, but it's going to just over the samples that Carolyn's generating, it's going to become much better, and then we're going to have more consistent names of these things, and it's going to, and then it'll link to an actual description page where you can go and say, if I, look at this 99 gene set, there's this link here. Right now, it just goes to PCBC, but it will go to an actual description page of this, and then show actually a heat map of this signature, so you can immediately get that, and then you output the table data of those expression values. And then another button in here we're going to have to most likely networks, although that may be best done with this a button that comes after this.
And one other thought I had is one thing that also might really help the adoption is if you could just find a couple of the PCBC researchers to really get into these tools and start using them and to pull together an interesting story of research and findings that they got out of using these tools that they could then give a presentation to all the other researchers, right? Like the phlegm to the water. That might be another.
Yeah, it's going to be one of our good guinea pigs, although they are focused on a finer point rather than some of the major points. But everybody's, it's always so much more interesting to take a specific problem at it. So if there was some group that was really interested in, DNA methylation, chromatin biology, that wanted to just make sure they understood really all of what you could put together from this, that would be fantastic. So anybody who wants to do a full-fledged project, we want to hear about, and
has joined the conference.
We would definitely pull out all stops to help them.
Yeah, I just think that trying to foster a couple of those in a sort of targeted way to get the traction going might be really helpful.
Yes. Another thing that I wanted to be like the beginnings of clear from this was the role of those differentiated lineages in really trying to understand developmental pathways and the potential of pluripotential cells to illuminate those pathways. It's just I can't say how important that is. It's really, I think that's what's going to make it just utterly spectacular.
Well, first again, this is Michael. I think what you're doing is important, and I want to encourage it, but I'd really like to see parallel tracks. One, that important work of yours. Two, the evangelism that Jenny just described. And then three, the basic training, because my perception of a lot of our fellows and junior faculty is that they're at the very, very far left-hand end of the learning curve here, and holding just to walk through where the buttons are and which ones to use when would mean a lot to them.
Yeah, I based on my experience, that was probably quite true. I would support all three of those.